So our focus for this set of videos is all going to be about protein synthesis, which is a fancy way of saying making proteins. If meiosis is all about how we pass our genes down to our offspring through the formation of gametes that will go through fertilization, protein synthesis is all about how we take our genes and turn them into traits. So that's going to be our focus, is talking about how we get from genes to traits through the formation of proteins. So the founding principle that kind of defines making a protein is what we call the central dogma. The central dogma is the flow of information in the cell. So we have information stored in our DNA about what our cells should look like. But DNA is a format that our cells can't use or read. It's like having an instruction manual for a piece of furniture written in Swedish. Most of you probably don't know how to read or write or understand Swedish, so you would have to take that instruction manual and convert it into a language you understood before you could actually make your piece of furniture. The same kind of thing has to happen in your cell. You have to take the instructions found in your DNA, you have to convert them to a language your cell can use, which we call RNA, before they can actually make that piece of furniture, in this case, our protein. So the flow of information is from DNA to RNA to protein, and that's what the central dogma is. It's this idea that information flows from DNA to RNA to protein. Now, the flow of information can only run in one direction. We can never go back from a protein, figure out the RNA strand, and work out the DNA strand from there. We can only move in this singular direction. Now, the first step to making a protein is what we call transcription. Transcription is the practice of taking DNA and converting it into RNA. This is what we call that process. So, here's how transcription works. Transcription occurs inside the nucleus, and it's going to be using the DNA strand. So the DNA strand, remember, is a double helix. And so if this is the gene we want to make a version of that we can read, because this is the instructions we need to understand, what we're going to have to do is uncoil the DNA and unzip the DNA so that we can actually get access to the nitrogen-containing bases. Once this is open, then our RNA nucleotides will come in and attach to the strand of DNA. They're going to use base pair rules, just like you learned with DNA, that DNA uses base pair rules to hold the two sides of the molecule together. RNA here is now going to use base pair rules for its own construction. In a very similar way to DNA replication, where we saw those nucleotides floating in and attaching, you're going to see the same kind of thing here with RNA. So these RNA nucleotides are going to come in and float in, and they're going to attach and begin to make a copy. And what's going to happen is an enzyme called RNA polymerase is going to move along the DNA and RNA strand and make sure those RNA nucleotides are bonded together. In a very similar way that DNA polymerase did during DNA replication, RNA polymerase is doing that same kind of function, making a solid strand of RNA while we are transcribing the DNA molecule. So here's what we get at the end of transcription. We have the DNA open, you can see. This is where we just left off. These here are the pieces of the RNA polymerase complex that have now broken apart because we no longer need them. And then here is our strand of RNA that we have produced. We call this mRNA, that stands for messenger RNA. And that's important to remember because we're going to talk about some other forms of RNA later on that you're going to need to understand the difference between this RNA and that RNA. So let's talk a little bit more about RNA so you can have a better understanding of what's going on. RNA is a single-stranded molecule. It's made of nucleotides, and these nucleotides are very similar to DNA's nucleotides with some very distinct differences. So we have the same phosphate group, and we have the same nitrogen-containing base for the most part. We have 4 nitrogen-containing bases in RNA. We have adenine, cytosine, and guanine, but instead of thymine, we have uracil. That's URACIL. Then we have a different type of sugar. DNA contains the sugar deoxyribose. RNA contains the sugar ribose. So this is what this looks like. This is the single-stranded molecule. And here you can see cytosine, guanine, adenine, and uracil all here present so that you can see the differences between this and DNA. So this is a comparison of the two molecules. So you can see this single helix versus a double helix. You can see the different nitrogen-containing bases. And of course, we talked about the difference being the sugar as well. So here we have ribose sugar, and here we have deoxyribose. So now we're going to talk about translation. Translation is the process where we take the RNA strand we made during transcription and use it to make proteins. So we're using that mRNA strand, and we're going to take it outside of the nucleus and use it to make a protein. So here's what that looks like. So our RNA strand has now left the nucleus, and then we have an organelle called a ribosome that bonds to the RNA strand, this mRNA strand, and it's going to be the center point of where translation happens. So it has 3 openings, and what's going to happen is this molecule called tRNA is going to come into the first opening here, and it's going to use these 3 nitrogen-containing bases called an anticodon to attach to the codon viewed here in this ribosome. So you can see these 3 pegs here. This is what this tRNA is going to attach to. And when it attaches to that, the amino acids that we're building here, this chain, is going to attach to this amino acid, and we're going to further extend the chain. And you can see as we move along, the amino acid chain gets longer and longer as we piece each amino acid into place. So this occurs outside the nucleus. Ribosomes attach to the RNA, specifically the mRNA. tRNA enters the ribosome and bonds to the mRNA and is linked, and it links the amino acids together to form peptide bonds, making a protein strand. So a ribosome is used for making proteins. It has a large subunit and a small subunit with 3 binding sites where the tRNAs are going to bind. So let's talk a little bit about tRNA. tRNA is a molecule that has an amino acid at one end and an anticodon at the other end. So this is a strand of RNA that's all folded up and has an amino acid group attached to it. This anticodon has a sequence of nitrogen-containing bases that will bond through base pair rules to attach to the mRNA strand. I'm running out of time for this video, so I'm going to stop here and pick up this conversation in the next video.